A very good morning, a very good afternoon, and good evening to everyone. Welcome to Asia PGI webinar series. And we are very grateful that you spend some time together with us to have these important discussions looking at can we enhance Nipah virus surveillance with wastewater and environmental surveillance. And we are very privileged to have our experts as panelists, Dr. Farah Ishtiaq from Tata Institute for Genetics and Society, Principal Scientist, who leads the Disease Ecology and Environmental Surveillance in TIGS. She has focused a lot of research effort in looking at the use of metagenomics, focusing on infectious diseases in environmental samples and wastewater samples. At the same time, we are also very grateful and delighted to have Dr. Erik Karlsson, head of Virology Unit, Institut Pasteur du Cambodge, where he leads the genomic surveillance of emerging viruses at the animal and human interfaces with a focus of avian influenza. He directs both the Cambodia National Influenza Center and the WHO H5 Reference Laboratory. With their expertise and experience, together with all the experts joining us for these discussions, we welcome you and we hope to seek more guidance and questions throughout these discussions. Please feel free to put in your questions on the FAQ if you have any, and we will try to capture and try to respond to these questions as much as we can during these 1-hour sessions. After the webinar, we will also try to capture some of these responses to your questions if we have not responded during the discussions after the webinar, and will be posted on our webpage. My name is Vincent at the Center for Outbreak Preparedness of Duke-NUS Medical School. Warm welcome again. As mentioned earlier, we will very much appreciate your input and guidance in the situation where you feel that wastewater environmental surveillance will be valuable to detect and enhance Nipah virus surveillance. Please let us know what will be the challenges you feel that any organizations or any authorities will face in the process of establishing this process. So you can scan the QR code to let us know your response, and we will try to share at the very end of these discussions with a summary of these challenges. I just stop for 1 minute, just to let you scan the QR code. In response to the recent Nipah virus outbreaks, this webinar aims to try to consolidate some of the needs and potentially resources from everyone here. And we are at the same time looking at exploring, establishing multi-sources, collaborative surveillance approach as part of the WHO advocacy as well. And the recent WHO webinar organized by CRO Office in partnership with WHO colleagues has been impressive and amazing. And thanks to their effort, we got to know more insights into the context, the situations in the recent outbreaks. So hopefully, to complement the WHO webinar, this discussion will be something very useful and potentially impactful for the region to enhance Nipah virus surveillance. So as part of Asia Pathogen Genomic Initiative, we aim to enhance WES in the region, in Asia. And as a first initiative, we have mapped wastewater surveillance activities in Asia. And through this living dashboard, this is what we have captured so far and the gaps that we have identified as listed in the box on the left through 81 projects in 15 countries. And if you are keen to find out more information, you can actually scan our QR code on our publications to find out more insights and results based on the data that have been contributed kindly, generously by our partners in Asia. Nevertheless, these are not exhaustive, and we highly encourage those who are new to this mapping exercise. Please feel free to scan a QR code on a living dashboard to contribute to what you have done or what you plan to do for wastewater surveillance in Asia. As a result from this mapping exercise, we managed to capture about 4% of the WEST project are actually testing Nipah viruses. And of course, these are mainly in India and in Malaysia, as much as based on the contributions from the mapping exercise. And of course, this may not be very accurate as we move along and encourage more partners to contribute to these living dashboards. So as part of this webinar, it is an important question to ourselves and to everyone is whether should we increase the number of projects, surveillance programs to look at Nipah viruses, including especially Nipah viruses. So as part of our Asia PGI integration use case framework for wastewater environmental surveillance, we advocate the need to ask the public health questions. So for Nipah virus, how can WEST actually contribute to educating, informing public health response and strategies, in particular looking at re-emerging Nipah virus, tracking potentially variants of Nipah virus, identifying high-risk settings, and also monitoring some of the public health intervention effects. And we also advocate the need to ask what will be the public health actions that can be taken by public health authorities timely, effectively, when some of the wastewater surveillance signals highlights the potential risk of transmissions of Nipah virus at the sampling sites. So could there be more intensified surveillance and quarantine among the human and animal populations? If the answer are yes, then the next question we need to ask ourselves is what will be the sampling strategies? Does the existing sewage system covers the population at risk, including the animal population at risk of Nipah virus transmission? Looking at potential contaminations from rainwater or industrial waste that might affect the integrity of detecting or sensitivity of detecting Nipah virus in wastewater and environmental samples. And very much in this discussion, we're going to have a lot more insights from Erik and Farah, looking at sampling strategies, detection, feasibilities. And then of course, if the answer is yes to the sampling strategies and detection feasibility, then we have to slowly progress to the next webinar that we try to understand about the sustainability of integrating WEST for Nipah virus. Just a quick reminder about the routes of Nipah virus transmission. Can it guide our sampling strategies? So as far as we know from now, based on evidence from research and outbreak investigations, there has been transmission of Nipah virus through the feces, saliva, and urine of bats, particularly fruit bats and flying foxes of the genus Bactopus, circulating the virus through some of the fruits that have been contaminated with the virus to the pigs that are nearby, eating some of these contaminated fruits, and therefore affecting the pig farmers, potentially the pig handlers, especially at the abattoir. And there has been also a number of different outbreaks as evidence to show human-to-human transmissions in Bangladesh, as well as from the raw contaminated dates palm sap that is consumed by some of the populations they are actually using and frequently consuming some of these sap itself. There have been also a number of different serological studies that look at potential transmission between dogs, horses, cats, that might be potentially also pets, to the human populations that might have caused some of these transmissions. The next question we can ask ourselves is, can we make use of the current outbreaks, locations, geographical regions, and the flying patterns of some of the fruit bats to guide our sampling strategies based on historical evidence? So with this, I would like to hand over the time to Dr. Erik Karlsson to share his perspective. Erik, please, followed by Farah. I'll stop sharing here. Over to you, Erik. Thank you.
Thanks, Vincent. Good to see everyone on the call. Hope everyone is doing well this afternoon or what time it might be for you. As Vincent said, my name is Erik Karlsson. I am the head of virology here at Institut Pasteur in Cambodia. And yes, we do a lot of avian influenza work, but we do a lot of other work at the bat-human interface. And so we have a long history of Nipah virus research and surveillance. I'm just gonna I wanted to just kind of give a quick oh, I think I opened up the wrong one. Sorry, all. Let me go back here. So I just wanted to give a quick overview of some of my thoughts on Nipah virus surveillance in wastewater, what we know, what we don't know, and then potentially, what we can do to improve it. So can you see that okay, Vincent?
Looks good. Thanks, Erik.
Okay. So really what I was going to talk about today is the feasibility of environmental and wastewater surveillance for Nipah virus. I'm not really going to talk about the virus per se. I am a virologist, but I'm not going to talk about the virus itself. But really, I'm going to talk about, environmental surveillance and how we might be able to use that for Nipah virus itself. And then Farah is going to give us a lot more perspective, I think, on actual surveillance for these viruses and more about the virus itself. So why Nipah virus actually challenges conventional surveillance? I think we understand that it remains one of the most consequential zoonotic pathogens, precisely because it really doesn't behave like viruses in most surveillance systems, right? So we're all used to doing surveillance for kind of, potentially predictable viruses. But in terms of Nipah, outbreaks are rare, they're highly focal, and they're often detected pretty late. So we also have to deal with a zoonotic transmission cycle that is technically environmentally mediated, right? So if we're talking about things that are contaminated with potential virus, and it's strongly shaped by ecology and human behavior. And I think my colleague Farah is going to talk a bit more about that later. But in context of the webinar today, what do we mean by environmental detection? So, I just wanted to kind of start with this and this basic aspect. So when we talk about environmental detection of these viruses, what are we talking about? So what we're talking about is, identifying Nipah virus RNA, basically outside of the host. And that can include things like water and wastewater, but also things like air and surfaces, soil and urine contaminated environments, and even contaminated food and food contact surfaces. As you heard Vincent say, there's been a lot of different ways that these viruses have actually spread between bats or pigs and humans. Importantly, and I want to bring this up when we're talking about this, when we detect something in the environment that does not equate necessarily to infectious virus, I think we have to be very careful about when we say we're detecting virus that it's infectious. It's not always true. Okay, just because we're detecting the RNA of a virus, it's not always infectious. And when we detect it in a water source or something like that, that does not necessarily mean there's a human infection that's ongoing at the current time, okay? So I think that those are really critical points here when we're talking about a One Health system. This is not just public health, it is animal health and it is environmental health at the same time. So in this context, environmental signals really are contextual, indicators of risk. It means the virus is present somewhere in the system, whether that be from the hosts or from a human infection or just in the environment itself. So I think we need to understand that environmental surveillance itself is really best understood as an upstream early warning layer. Okay, it is not a clinical diagnosis. So in terms of the virus itself, and I wanted to bring this up because it's really important, how long does this virus actually last in the environment? Because that actually allows us to be able to detect it. So Nipah viruses, so the family that Nipah belongs to, are pretty environmentally fragile, right? Not instantly so, but they are pretty fragile viruses. We know from evidence that these viruses can last several days in things like pH neutral bat urine, sugary fruit matrices, which are, really on fruit and things like that, or contaminated sap. But anytime they're exposed to things like heat or desiccation or UV or even extremes of pH, they fall apart pretty quickly. So, environmental detection windows for these are actually pretty short. So to have a meaningful, understanding of these viruses in the environment, we have to act pretty quickly. In terms of what we can look for, and if we start in sort of a public health manner, where could we actually look for these viruses to actually give us some information of what's going on? First of all, I know that Asia PGI and many other people really focus on things like municipal and facility wastewater. So I wanted to talk about these things. The best settings likely are going to be places like hospitals and referral centers where, potentially cases are going. Things like encephalitis wards and isolation units. But really, we need to focus on small, defined catchments. Okay. As I said, and as I think Farah will talk about, these viruses are really happening in small spillover events. They're transmitting from human to human, likely in hospital settings, right? So really, we need to instead of these big broad catchments, we may need to go to smaller catchment areas. And we may need to be focusing on things like livestock interfaces, so like pig farms, slaughter facilities, things like that, instead of, say, municipal areas. Why that could work? Obviously, Nipah is shed in urine and saliva and feces. But these wastewater could integrate signals from symptomatic and pre-symptomatic individuals. From the literature, we know that these individuals are potentially shedding virus for up to 20 days, which is quite long. And so we don't really know a lot, and maybe Farah can expound on this a bit more, is about the asymptomatic period versus symptomatic period and things like that. But really, we need to be able to understand spillover and amplification risk. Another thing that potentially places are looking at, a lot of airports are on alert at the moment here in Asia, is aircraft wastewater. So basically, can we use aircraft wastewater, which we know has been used successfully for things like COVID-19? Could we actually use it for capturing signals from travelers before they actually contact healthcare? So could it provide us some early indication of cross-border movement of a potentially infected individual? And it's technically non-invasive and independent of testing compliance. Basically, at the current time, targeting sampling of flights from areas with known or suspected Nipah activity would be ideal if we want to catch this, but it's really requires some situational awareness during regional outbreaks because it's not like a seasonal virus where we can look at, loads of things moving across borders. It's really going to be an isolated incident, and it would require a lot of active surveillance, which we all know is pretty, intensive for labor and cost and time. So it's really a consideration that a country would have to make to be really focused on this overall. And what I wanted to talk about today a little bit more is really what do we need to detect Nipah in environmental samples? Because I think this is a critical point for everything that we're talking about. And I think there's a few key requirements. First is a highly sensitive lineage inclusive RT-PCR. Okay, like if we're going to be able to detect these viruses successfully in all places, we need to have a highly sensitive PCR. We need to have careful interpretation. Our detection is not probabilistic. It's not diagnostic. And then, of course, this is Asia PGI, right? We want to have sequencing confirmation and genomic epidemiology as fast as possible when the viral load permits to be able to do things like understanding where that virus is coming from, if it's mutating, how it's spreading, and things like that. So for me, these are the key requirements we might need from environmental samples. Just to touch on this briefly, in case you don't know, Nipah has some genetic diversity and why lineage matters, okay? There's two major lineages of Nipah, what I call lineage 1 and lineage 2. Again, that does not imply anything other than there's two distinct lineages of this virus. Oftentimes, it's considered the Malaysian lineage and Bangladesh lineage, but we don't like talking about certain place names, so we're not going to call them that, okay? Lineage 1 is historically associated with pig amplification and things like that, whereas lineage 2 is more frequently linked to direct bat-to-human transmission and recent documented human-to-human transmission in nosocomial settings, so in hospital settings where people are in close contact with that patient. Evidence indicates that both of these lineages circulate in places like Cambodia. So here where I work, I find both of these lineages, whereas in other places, they may have one lineage or another. And so environmental and wastewater assays, especially for testing from airline surveillance or in places like Cambodia where we have both lineages circulating, they have to be lineage inclusive. If you look at something like avian influenza, there's a lot of avian influenza PCRs out there, but a lot of them are made for, a continent or a country-specific purpose, and then they can't detect the viruses that are circulating in another country. Okay, Nipah is the same thing. There's a lot of PCRs out there, but you have to validate that they work for both lineages of the virus. Okay, and we have to avoid things like primer bias and sensitivity across genetic diversity. Sensitivity itself is foundational. Okay, as we've said, and as I think Farah will talk about, these animals, these bats, do not shed a lot of virus into the environment at one time. Okay, it's often very, very faint and very low concentration. So we really need highly sensitive PCR assays and careful management of inhibitors so that we can actually detect this virus. For example, in the assay that I use in my lab, which is an in-house assay. Again, this is not the WOA assay. This is not the WHO assay. It is the one that we use in my laboratory that is generated in-house. We know that we're capable of detecting down to 5 to 50 genomic copies per reaction, which is pretty good sensitivity. And it's validated on both the Bangladesh and Malaysia lineages, okay? So I know that my assay in-house can be used down to a relatively good sensitivity, and we can use it against both lineages. I've put it up here. It is freely available on protocols.io for anyone to use, including our primer and probe sequences. We will share, I guess, a PDF copy of this later if we can. The other thing that we have to think about is sequencing. So how do we confirm and contextualize that detection? Often these days, PCR alone is not sufficient. Governments and other people always want to know more about what you're detecting, whether that be, the context of what you're detecting. If it's say in the case of Cambodia, they're going to really want to know whether or not it's a locally circulating strain versus something that may be imported, right? And these are only things that we can tell with generating sequence. So sequencing really allows us to confirm our specificity. It looks at lineage and it really monitors viral evolution. We know that there's amplicon-based systems for Nipah that work well. And unfortunately, reliable results typically rely on CT values that are below 29 to 30. That may be very difficult in environmental samples. And unfortunately, since this is a BSL-4 level pathogen, it's quite hard to isolate the virus to get sequenced. So, samples that are above 29 to 30 CTs in our hand, at least for N gene, typically give incomplete genomes, but you can still get contextual information from them. So I do encourage you to keep sequencing. And you really need to build your sequencing capacity into your surveillance design from the start. Okay, so you should have conversations with your ministries and with your laboratories that whenever something is detected positive by PCR, that sample needs to get to somewhere that can actually sequence those samples pretty quickly. And there's a lot of labs that are able to do that in many countries. In my lab, we have developed an amplicon-based system. Again, that's validated against both Bangladesh and Malaysian lineages. And again, that's freely available online at protocols.io. And you're welcome to use it here or talk about it with us later as well. The other thing I just wanted to touch on, and one thing that we do, throughout what we do here at Asia PGI and partnerships with Vincent and other people, is environmental metagenomics. We use environmental metagenomics a lot in my laboratory in a lot of high-risk interfaces like live bird markets, swine abattoirs, but we also use it at bat-human interfaces. We like it because it provides that unbiased detection, and we can actually detect unexpected or novel viruses and insights into broader viral ecology. In our hands, we have been able to detect Nipah virus reads in bat urine as well as environmental samples, things like air and water from bat roosts. However, the signal is typically very, very low and reads are very sparse. So we don't get a lot of full genomes. So when we do this, it would really require a lot more active surveillance and really monitoring these populations, and we're working on it. And detection tends to be inconsistent. And I think Farah will talk about that a little bit, why, in terms of the ecology of these viruses. But it is excellent for discovery and contextualization and hypothesis generation. So, I really do encourage this. If you have these samples, you want to see if you might have Nipah in a large pool of samples, you run some metagenomics on it, you see if you get a Nipah hit, and then you go in with some more contextual PCRs and things like that. Again, best used in combination with PCR. PCR is your gold standard. Remember that up front. So what do we know from the literature about being able to detect this in environmental samples? In terms of documented detections in these samples, obviously raw date palm sap, sap collection vessels, but also we have been able to find it in places like surface water beneath bat roosts. Sorry, I'm losing my voice, everyone. In Cambodia and Malaysia, which oftentimes may be contaminated by urine or feces or other things from those animals. And we've been able to find it at standing water at bat roost sites. So when we do environmental sampling, which is not necessarily surface water, this is standing water. And then there has been some evidence of water in pastures contaminated by bat excreta in Australia for Hendra virus, which again is a cousin of Nipah virus. So there are documented examples of this in the literature. However, when we talk about wastewater environmental sampling, I think it's very biologically plausible and something that you could go out and do, where you look at farm and farm water and runoff, drainage water from animal holding and slaughter areas, as well as human wastewater and mixed human livestock wastewater systems to be able to detect this virus. So I do encourage people to try to look for these things. We certainly are trying to do the same. And hopefully as a community of practice, we can share what we're finding and really understand how we can use our wastewater systems and other things to respond to Nipah. And the question now, of course, is, will it work? And I hope that we'll all work together to actually find out how we can better detect Nipah and use these techniques for not only surveillance at these high-risk interfaces, but also potentially for cases that we may miss within healthcare systems. So with that, I'll just say thank you to everyone we work with, especially Asia PGI and our funders at Asia PGI. And I will stop here and hand it back to Vincent. Thank you very much.
Thank you so much, Erik. So much of insights, very, very precious and very, very helpful. Without further ado, I would like to invite Farah to share her perspective. Feel free, and you can also provide additional comments to what Erik has highlighted. Over to you, Farah. Thank you.
Yeah, great. Thanks, Vincent. And I do not have any slide, to be honest. I would just talk through some of the points which Erik has mentioned, but also really want everybody to kind of think of the whole scenario from an ecology perspective as well. So there are two aspects of this Nipah outbreaks. There is one is an environmental drivers, but also there is a social or a behavioral driver which plays into that. And I just want everybody to step back and just come out of the whole virus thing, but think about the hosts which are spreading, these viruses and how this is happening. So Nipah virus, I mean, is the same group of virus which actually falls into the Hendra virus, which with the outbreaks have reported in Australia, and the same transmission happens, from the change in the land use pattern at the time when they changed the savanna to agricultural fields. So the habitat became so restricted that these bats start to get more in contact with these horses, which became an amplifying host. And when the humans came into contact, this human to human transmission happens. And then soon after that, these outbreaks of Nipah virus were reported from Malaysia and as well as in Singapore back in 2001 as well. Now, all these Nipah virus or Hendra virus, and even the Ebola virus, actually, they are transmitted by these fruit bats, basically, who like eating. With these bats, they thrive and they eat basically fruits or any sweet. And in our case, there's also the date palm plantations where they really thrive. That's like their favorite fruit. Now, about these bats, I mean, if you really are trying to find a bat infected with the Nipah virus, it's really hard to find one actually infected with, especially when they are in a big roost shedding the virus. However, in a colony of those bats, when they are roosting, or there could be one or two really shedding the virus. So basically, this shedding of virus, as one of the best bat scientists who studies the virology, he describes it, is a bit like Christmas lights, Something might be, one or the other individuals might be shedding at a time. So when they are roosting together in a very big colony, they come in contact through their urine or feces, and that's where these contact rate increases and other individuals are constantly getting exposed. So if you look for the seroprevalence in a colony of these viruses, you do adults and also the sub-adults, the pups also being exposed to these viruses. So the shedding can be happening any time of the year. But however, there is a very stark seasonality in terms of the humans, like when they are getting in contact with these viruses. And that actually usually happens during the time of December to April. And that's the time which is where it is coinciding, at least in most of the Indian context, I would say in West Bengal or even in Bangladesh as well, where people tend to extract these date palm sap where the bats probably have fed into or maybe have urinated also, and when they have these high concentration of viruses. Having said that, when they are exposed and they are drinking the sap, they are getting in contact, and that's where the transmission is happening with the virus. So there is a kind of seasonality from the environmental point of view. That's where it plays out. Now, it is true that it's not necessarily through humans as well. It could be the pets or the pigs or other reservoir hosts, which could be playing a role where humans are getting constantly in contact with, and especially in a very high dense area or areas which are very disturbed, where you could be having satellite routes for these viruses, bats as well. Now, in terms of the environmental surveying, these are a very, I would say, the hotspot sites, if one try to understand where these roosts are. And I think that could be the probabilistic sites where one should be concentrating on, because where I'm sitting right now in Bangalore, we do have lots of roosts. I mean, I see that's the light, in my balcony, like 3 meters from where I'm sitting in my living room. They come and they are feeding on these for the nectar on other flowering fruits here and there. So basically, in a city like where we are, the boundaries are kind of blurred. That not everything is happening in the in a rural area or in an agriculture field. It can be within the city where those habitat earlier used to be the rural areas, which have now been converted into apartment buildings, or a lot of massive land change has happened. now these bird colonies have been disturbed, and now they kind of find these satellite routes. So people can come in contact with that. And this is where I feel the sewage surveillance that we've been conducted for the last year have been really interesting for us from the point of this pandemic preparedness. What do you see and what can you, what you can't, which actually Erik has very beautifully described that, yes, the gold standard is a PCR, but you also combine it with the metagenomics also for the whole city sewage, but also from the other satellite sites where some of the roofs have been there. But from the city water, even though we see lots of, say, for example, influenza viruses associated with human influenza, which is the seasonal influenza, but also the avian influenza viruses, but we have never seen any Nipah virus to date. And I think this kind of boils down to the fact that the viral shedding for the Nipah virus is extremely low from the bat's point of view. And even if you have human cases, I mean, which could be, say, misidentified, say, encephalitis cases, or people not reporting, say, subclinical, is also very low, probably. That's why finding them in the wastewater can be very challenging. But at the same time, I think doing a longitudinal surveillance to understand what are you seeing, recording a signal, I think makes it a very important case. And also looking at a community level as well. So for example, in some of the studies which have been done in Bangladesh, where the Nipah virus outbreaks have reported, and also across the sites where the bat roosts are there, they have not just been looking into the viral shedding for the bats, but they also have been looking for the seroprevalence in the other part of the community, like in the dogs or in the pigs or on other animals, which could be the reservoir host. So from that point of view, I feel those sites can be surveilled as well for looking at the environmental samples, as Erik rightly pointed out, through the standing water or areas where other animals are coming in contact, just to understand what do you see in terms of the Nipah virus. Can it be detected, or if it's been detected, can there be any hidden cases in those environment, in those areas? So yes, it's a part of the whole our environmental and understanding the whole behavioral aspect as well. But actually, it needs both the kind of surveillance tool, both environmental, but also changing the kind of behavior that perhaps not drinking or covering those, the date palm saps with some cover, so it's not directly contact with the rats, or also these bats can minimize some of these spillover events that can be looked into, because surveillance is a part of the thing which can help you to prepare and understand where the outbreak could or might be happening or have happened. But actually minimizing the risk or understanding these drivers, I think, is equally important. So these spillover events can be minimized to a certain extent. Vincent, I think I'm going to stop here, and we can take questions and more points for discussion going forward. Thanks.
Thank you. Thank you so much, Farah, for the insightful perspective as well. In addition to what Erik has provided as the initial insights. Maybe let's try to go to the Q&A. And I think there are some questions that we may need, your insights as well. So, there's these questions about, do you have experience using a concentration extraction identification device to detect viruses? If so, what were the results? Is it more applicable to remote environments? So I'm not sure whether Farah, Erik, if you have heard about this device, which is quite an interesting question. Yeah.
You mean a device to extract RNA from any viruses, is it? Is that what it refers to?
Yeah.
Yeah.
It may sound like more like a point of care tool to me.
Mm-hmm.
So
sorry, Vincent.
Yeah.
Repeat the question one more time.
So do you have experience using a concentration extraction identification device? In my impression, it sounds like a point of sight tool to detect viruses.
You mean in the remote area, is it?
Yeah, especially in remote settings. And if we have tried before, or if you have tried before, how is the experience, and what were the results? Just feel free to add on. Farah, Erik.
Erik,
Or
do you want
even
to
you
take
can
it?
use a use case from avian influenza, or the current existing wastewater surveillance programs in India.
I mean, yes, I mean, we do collect a lot of, environmental samples, not just the wastewater or the standing water or even the soil or fecal samples in remote areas. And usually they are either stored in a medium or in a really low, temperature, and we bring them back, say, within the 4 or 5 hours back to the lab, and then that's when the whole process actually starts. And there are a lot of new innovative ways now people are coming forward where they are designing these assays where you can preserve the sample in the field itself, which minimize the degradation of these viruses, because these
Exactly.
which actually helps to stabilize the sample a bit for longer, which can be then dealt with in the lab setting. Yeah. And Erik, you have your own experiences. You've been working in so many remote areas.
Sorry, I think my Wi-Fi may have cut out. Vincent, can you just repeat the question?
Yeah, so the question is, do you have any experience using a concentration extraction identification device? So it's like a, lab on the chip kind of a concept. I mean, as far as I can understand from my experience working with you, Erik, is that, the mobile labs could be potentially another platforms that this question is trying to refer to. And how was the experience? What were the results? Was there any challenges? And I guess the background to this question is about, having the need to preserve the integrity of the samples because of the potential long distance of travel between collection sites to the laboratories, may be a crucial factor for better performance of detection. So over to you, Erik, based on your experience.
Sure. A lot of what we do in my laboratory is to make things faster, better, cheaper, and able to go out into the field. So we get a lot of what I would consider toys sent to us by rather different companies, mainly to take out in the field, in the mud, in the rain, in the 45-degree heat, and see if they work. I have not seen an individual piece of equipment that you can just put a sample in and it, concentrates it, extracts it, and runs a PCR. But that would be super nice. I know that there's a lot of handheld devices that use LAMP and other technologies that allow you to take a sample and kind of extract in the field with a syringe and stick it into the device and get a LAMP PCR going. Those work relatively well for things like avian influenza. But again, those are mostly closed systems. So I highly recommend that you avoid closed systems where you're buying necessarily cartridges from a company without you validating that it will work on the virus that you have in your country. Because we found a lot of times, like in the case of avian influenza, if we buy a European test that they're making certain tests for in the European context, they don't work here in Southeast Asia, right? Because the viruses we have here are circulating completely differently than the ones in Europe. And obviously, the companies are making for their for their main stakeholders. So be very careful when you use these commercial tests. I'm not telling you don't. I think they're very, very useful tools, and they're very good for portability and ability to get into remote environments. Just make sure that it works on the strain that you're looking for, because you don't want a false negative when you're out in the field, 12 hours from the nearest laboratory. Another thing, as Farah indicated, is there's a lot more medias and ways that you can collect samples to get them back to your laboratory. Obviously, this is a BSL-4 level pathogen. You really do want to inactivate samples, preferably in the field if you're going to take them in the field, because a lot of people don't have BSL-4 level or even BSL-3 level laboratories to be able to work on these viruses. So, there's inactivating buffers, there's field extraction kits and things like that. And then what we use a lot now and what we've moved towards is coupling things like field extraction. So there's different types of field extraction methodologies with chips and with small machines, with pan-viral PCRs. So we do have a Nipah pan-viral primer set that we can actually run within 5 hours in the field that then goes directly into a nanopore sequencer. But again, that's not something that's hands-off from sample to sequence. You do have to move things over, but they are viable to use, very far from civilization, as it were, on battery packs and things like that. And they can give you quickly, down to the species level, a 600 base pair or 800 base pair sequence from that virus that you're looking for. So these things are viable in the field.
Thanks so much, Erik, for the additional useful information. Farah, will you have any further insights to add on? If not, we can move on to the next question. I think you're muted, Farah.
Sorry. Yeah, we can move on to the next question. Yeah.
Thank you. So I think this question is also, the upcoming question is also very important, and also something that is debatable, especially when we want to build a more sustainable Nipah virus surveillance through wastewater and or environmental sampling. So the question is, for the surveillance design, especially for environmental surveillance, is only once a year sampling potentially sufficient? Potentially, the question is looking at seasonal approach or more targeted location approach, or based on the flight path of the bats or the at-risk populations locations, or example in slaughterhouses, livestock farms, and primary care medical centers. This question is also with the background that there's only so limited labs. Potentially BSL-4 labs is able to test the Nipah virus, and is it something that is feasible for a limited lab capacity to do more frequent sampling and testing? Over to you, Farah, Erik. Maybe over to Farah first.
Yeah. Thanks, Vincent. So yeah, I think this is a very very important question. I think one of the reasons, I would say, from Indian context, the way we handled, say, even SARS-CoV-2 so well, that it was not just the testing was just not restricted to only few government labs. There were so many labs or so many points opened up everywhere that a lot of institutes and hospitals and everywhere people were involved, and they could test and eliminate, and contain the infection. And I think the similar approach needs to be applied for any pandemic pathogen. And that's where I think environmental surveillance kind of makes it slightly easier, where if you do not want to touch maybe really a bat or a human, but you can actually look for environmental signatures in the environment, and you can test those in some designated labs, and you can have a network of lab where these kind of work can be done. And the second aspect is that once a year surveillance, I don't think is very meaningful. I think it requires a very consistent and regular testing in a way that you can give a kind of your finger on the pulse, as I call it, where like even today, nobody is showing any sign of, or people are vaccinated for SARS-CoV-2, but we still look for that signal in the wastewater to understand, you have people shedding the virus, and if there is a rise in signal, what variant might be driving? Is it a low path, or what, or are we up against something new which is coming up? And I think similar applies to here, that what are those geographies in the context of where people are sitting, that and also not to do actually alone. I think having a map of where the bats are, where are people actually getting infected with, where the outbreaks have happened. I think that background information is also very, very important. And that's where I think this whole One Health approach comes into play, that we might be doing this work in our labs as a virus person or a molecular biologist, but I think you still need those people who are working out in the field and ecology and doing these surveillance, who just from the bat seasonality and understanding what they feed on, to getting that basic primary data. So, and understanding how the land cover is changing the patterns of these distribution patterns of these bats, because a lot of shedding actually happens in these bats is because they are lots of environmental stressors, which drives these scenarios. So it's a kind of a very mixed, kind of a very complex scenario here. So I think so these environmental surveillance is just not a standalone thing. It needs a very thoughtful approach, and I think it would work beautifully if it's taken into consideration using these points in. Yeah, I think Erik, you have more to add. Please do.
Thanks, Farah. About the need to emphasize for multi-sectorial, consultations and inputs and contributions, to try to understand the best sampling strategies.
Yeah.
In a more sustainable way. Yeah.
Yeah.
Erik, over to you. You have any additional?
Yeah. All early warning and surveillance activities require active surveillance. So when you say once per year, I would not, from my perspective, recommend going out once per year. I understand that these things are costly and hard, but to be able to understand signals as Farah and I have seen, especially with different, seasonalities of the bats, whether it's the fruiting season or, it's mating season or it's whelping season, these are really important critical time frames to understand the persistence of the virus in the environment. So, you really need to get it as granular as possible, getting out to these sites and taking regular samples. And the better you can do that, the faster and cheaper you can do that, the better you will be to be able to detect a signal before it becomes an issue in the long run. So when you're talking about sampling strategies, obviously you maximize based on your budget and your sustainability, but you want to be able to get the best baseline possible so that when you do detect a signal, you can react very quickly.
Totally agree on the need for a bit more frequent sampling strategies to create that baseline and to be able to set up potentially some threshold based on the baseline. Right. Thanks, Erik, so much. If I may just refer you back to our slides on challenges, just to see like what has been shown there as our last 5 minutes of discussions. Can you see the slides?
Yes.
So thanks so much, everyone, for contributing to these questions, focusing on the challenges to detect Nipah virus with West, based on your experience, based on expertise, and I can see a very big proportion of you, everyone here, contributing to biosafety as a key challenges, funding, sample, as we have just discussed earlier, based on the previous questions, sensitivity as well as laboratory, country sharing. In terms of sharing, as Erik has mentioned a number of times, the concentration or the shedding load potentially is very, very low, and therefore he recommended the need for metagenomics approach, monitor risk. So over to you, Erik and Farah. Will you have any specific challenges that you just pick one that you may want to comment a bit more with the last 1 minute for both of you? And potential solutions, if you have any recommendations. Yeah.
I think I would just talk from the Indian context where we are. I mean, we have a lot of cities who are involved in wastewater surveillance for the last 4-5 years, and they have moved on from SARS-CoV-2, say other influenza viruses, and looking for AMR and other things. And I think it's high time we really think about where to find sites where other pathogens of pandemic potential are. Like, for example, avian influenza is something which has surfaced in now, and we are trying to develop protocols towards it. But similar, we should be looking for what are those sites one should be doing for Nipah as well, if it can be kind of contained or understood better, where the spread is. And also, as I said again and again, that I think combining it with some real ecology studies to understand the roosting pattern, where the roosts are, where the bat roosts are, how it interacts with our environment and other larger community, not just humans, but other animals. I think those studies are very limited. And I think that would go a long way if we combine them with some basic surveillance to really tap into this problem of where the outbreaks will happen and how to contain them. Yeah.
Thanks, Farah.
Mm.
Erik?
Yeah, I think actually, Vincent, I would like to address one of these comments in the Q&A for my
Sure.
final comment. And it's the final question about, you know, how does environmental surveillance and other factors actually come into a proactive framework? And you know, what other things can we do? This person is anonymous. I won't call them out, but you know, if we can't really detect infectious virus, we don't really know that something in the environment is infectious or anything like that. You know, how do we prevent spillover, right?
Yeah.
And we have to remember that environmental surveillance as a whole is requires a paradigm shift in the way that we're looking at things. We are not looking at basic prevalence. We are not looking at outbreak response. We are really looking for high-risk areas where these things might occur. So when we talk about how we prevent spillover, in my opinion, we use environmental surveillance to identify times, places, you know, potential routes of transmission that could potentially lead to spillover. And then we do things like, you know, social dimensions of human interaction, as this person says, which is, you know, the ways that people interact with the environment, interact with agriculture, interact with their animals and wild animals, as a means of preventing that spillover, right? So it allows us to target those interventional activities and the risk comms and all of that kind of stuff to the right places, to the right people, so that we can so that we can use the social and the economic dimensions to actually prevent that spillover in the first place. So I think they're not mutually exclusive. They have to go hand in hand with one another, but you can use environmental surveillance really, really well to target what you're doing and actually optimize that spillover prevention, because not every pig farm is going to have a Nipah virus, you know, signal, but the ones that do really need that intervention first, right? So I think that's sort of what I'd like to leave off on this webinar as well.
Thank you. Thanks so much, Erik. And I totally agree in the sense that the interfaces between the animal and human will be, you know, one of the top priority for, you know, potentially some of the environmental samples to be collected for a more frequent sampling as well, just to have better assessments and earlier signal of potential spillover from, you know, the pigs to or animals to human populations. But of course, having said that, I guess on the ground itself, technically or you know, feasibility-wise, you know, there's a lot of negotiations and discussions will be needed to be conducted with potentially the pig farmers. And what is the actions after the signal has been found on the pig farms? And would there be a question of whether is there a need to count all of them, or is like, you know, how do we quarantine some of them and monitor the situations, or do we need to count them straight away? Based on the experience from Malaysia outbreaks previously in 1998 and 1999, I think they have significantly, you know, suffered quite a huge economic loss due to the culling of, you know, many pigs. Yeah. So it's still a question whether that's something feasible on the ground. Yeah, even though it's one of the top priority for samplings. Yeah. So I know there are actually more and more questions coming in, and I have to apologize that we will not be able to address all the questions here, but definitely, you know, we'll be working very closely with Farah and Erik to look at the questions, and we'll try to respond as quickly as possible to your questions, and we'll post it on our Asia PGI Center for Outbreak Preparedness webpage, you know, after this webinar. Please give us a bit of time. I know this information may be very important to you to think about and to consider, you know, a bit more on establishing, you know, collecting wastewater or environmental samples, you know, to detect and enhance Nipah virus surveillance. And with this, I would like to thank again very much to all of you for attending these discussions. And we will try to share the slides and you know, the video, the recording of the discussions. And definitely, last but not least, thanks to Farah, Erik, and as well as my colleague Ashley here for supporting us in generating this very informative, hopefully to some extent conclusive, but definitely there are more questions that we need to address as we debate and discuss over the need to establish and sustain wastewater surveillance for enhancing Nipah virus transmissions surveillance. Thank you, everyone. Hope you have enjoyed, and we'll keep in touch. Thank you.
Thank you.
Take care. Bye.
Bye.